Hi everyone, my name is Ramkumar Mathur. I'm assistant professor at Department of Geriatrics, School of Medicine, University of North Dakota. So my lab is basically looking role of tumor permissive environment and how they are important in the aging colon cancer. So in particular, we were interested to understand mTOR signaling, how they modulate in aging colon cancer, and they can make like more metastasis and like made more like the death in the aging people. And so today I'm going to present the work which is funded by DaCCoTA Feasibility Award. And the title of my talk is Regulation of mTOR Signaling in Colon Cancer Pathology. So as you can see, like colon cancer is a colorectal cancer is a like the leading cause of cancer cell death in the United States. More than 140,000 people annually they diagnosed with this colorectal cancer, and around 50,000 people get like die from the colon cancer. So this is like really striking and indicating an urgent need to find effective treatment strategy for colon cancer. So basically, colon cancer is a start in the colon or on in the rectum with abnormal cell growth. But most abnormal cell growth is not like cancer. It's called like colon polyps. However, few of abnormal cell growth can turn to malignant and become like tumors, and this tumor has the capacity to invade to different tissues and is called like metastasis, and the studies has shown that metastasis is really like is critical to like pathogenicity of the cancer and like the more like the risk for like the cancer related cell death. And in surprisingly, you can see like the aging people has a like high incident rate for the cancer, colon cancer, as well as like they have like high death rate compared to the young people. Like you can see like the aging people has like the really like the highly increasing death rate. So which indicate that since like the aging people has like the uh, down-regulated immune system, dysregulated immune system, complex gut environment, which could be like the provide the tumor permissive environment like different than young people, and then they have like more metastasis, and then this may be like the leading reason for the they are like the incidences like the of other dying is like more so to find out that what is the like mechanism of this like the tumor permissive environment, so we look at the mTOR pathway, and the mTOR pathway is actually a very well known and in the cell growth and the cell survival. But however, this mTOR dysregulation can cause cancer. As you can see, like with the mutation with the mTOR, it can increase the mTOR related pathway. Uh, Also, like if you see like the hyper mTOR signaling can cause like the accelerate the aging. So which actually make the more connection with the mTOR and like increase mTOR and associated with cancer and increase the hyper mTOR signaling is like also like the causing like more aging. So which could be like the mTOR is a common mediator to regulate the cancer pathology in the aging people. So based on that, like the we hypothesize, and then we make like a two different aim of the study, and the first to investigate the role of mTOR's function in tumor permissive environment, and second, can we block the mTOR activity, and then to reduce the colon cancer in the older adult. To test that, we get the colon cancer biopsy from the pre-archive samples, and we look at the fucosylation process, like the which is like the fucosylation process is the post-translational modification of adding the oligosaccharide or like the sugars to the protein, and this as you can see like the classical example when the this TJ beta receptor, which is well known, and this metastasis and the fucosylation of TJ beta receptor with the fucosyl transferase 8 this like enzyme, this helped to the fucosylation, and this epithelial cells leads to epithelial mesenchymal transformation, and which cause like more migration, invasion, and metastasis. Inhibition of fucosal transfer rate can inhibit the metastasis. As you can see that. So surprisingly, we find that the old colon has like on a steady state they have more of fucosylation, as you can see, like more UEA and stain, and the WJ, which stand for the fucosylation, and this is trending indeed like really high, suggesting that increasing fucosylation in the aging condition may have a link with the higher risk of colon cancer in the old people. So to dash that, like we look at the this aging condition and the cancer condition, then we found like indeed like the old people colon biopsy shows like a increased fucosylation as compared to the young people, as you can see over here. And with the we checked in the different stages of the cancer, suggesting the increased gut fucosylation is associated with the high risk of colon cancer in the older adult. So later on, then we look at the single cell RNA-seq in the colon patient, and then we found that the epithelial cell has really increased fucosylation in the gut of the this single cell RNA-seq, and then we found interestingly like the old people, the normal state like steady state has like a more fucosylation, as well as like the tumor condition, the the IL-22 receptor is is the one like which is like involved. People have shown like IL-22 can induce like more fucosylation, and this is like really very striking like a link that IL-22 is like receptor is like really high in the tumor condition compared to the young, so suggesting that maybe like old people has like higher IL-22 receptor signaling, which is like the downstream like induce more fucosal uh, few two expression and linked with a more tumor permissive environment. So then we look at like whether IL-22 can induce more tumor fucosylation, this fucosal transferase, and then we incubate the human recombinant IL-22 to epithelial colon cancer cell line, and then we found that increasing dose of the IL-22 can increase TGF-beta receptor, which is cause like the tumorigenesis, to feed it, and increase the more tumor permissive environment with more higher transferase, fucosylation, and like as well as the TGF-beta receptor signaling. Also, we look at the IL-22 downstream mTOR pathway, which are detected by phospho S6, phospho P70, and which are like inhibited by rapamycin, which is the inhibitor of the mTOR, suggesting the IL-22 receptor can induce mTOR signaling and downstream for 2 and FU2 and FU8 pathway, which are responsible to make like the more permissive environment in the aging colon cancer condition. Uh, so later on, then we block this the resveratrol, which is uh, anti-aging compound or like senescent inhibitor, and which also has like shown capacity to inhibit the mTOR activity. So we what we did, we look at the cell migration assay in the DLD1 cells, and then these cells we found the incubation of the resveratrol actually like restore the minimize the cell migration, as you can see here, and as well as like the this resveratrol decrease the FUT2 and FUT8, suggesting like role of FUT2 and FUT8 in the cell migration of the DLD1 cells. This is the like the in vitro assay, and to validate the blocking of mTOR to elevate in the colon cancer, and then we look at the mouse model, and then tumor model like just find like direct evidence is that. So we look at the young and old mice like as like human condition, like the old mice has colon has like more fucosylation compared to the young, which we validate with also like the flow cytometry like staining. Then we breed the mice with in the it's called a TSC, which is like negative regulator of the mTOR. And then if you take it out the TSC, then mTOR signaling is really high. So we expect that this mice may have like more fucosylation and like increase or hypersusceptibility to get uh, colon cancer. So we decided to go ahead with the DSS model, and then we now this these are like propose like this experiment is like we are like this like going to test this like in the upcoming times. And then I hope like that we can finish like this experiment like soon to just to validate whether, and we look at the fecal solution how the I mean look at the we will look at the more tumor permissive environment, how this like mTOR signaling, which will like direct evidence and like in the fecal solution as well. So basically, the working model we have here the IL-22, which is secreted by particularly in native blood cells, and this IL-22 can actually in the is in, increase the fucosylation, and then aging condition has the more IL-22 receptor as we see, and the aging tumor condition also promote more IL-22 receptor and TGF-beta receptor, which actually make more tumor permissive environment. And fucosylation is like really like make that more provide like more promoting like tumor promoting environment. And then mTOR is, which is like the downstream of this IL-22 receptor, promoting like more feedback loop to promote like this in aging condition. I would like to stop here, and I would like to acknowledge my lab members like and DaCCoTA feasibility grant. Without that, this work is like really impossible. And co-PI on this grant to Dr. Donald Gilbert, Chair Genetics, and Dr. Otis is the oncosurgeon. So we collaborate Dr. Stika, then we will get like more human samples, and then we can test like more validate our data with the human samples and as well as the mouse like that. So I hope like we can like the finish that work soon as well. I would like to uh, uh, acknowledge UND Imaging Core, Histology Core, Flow Cytometry Core, like always helpful. And the outcome of the grant, like the we just like the this work the shown I work, the work I shown that that is like the currently in the revision in JCI Insight. And the also like the using this data like the one of my student like he got like so Crohn's colitis foundation. And we are like the currently in the resubmitting the NIHR1 like so I hope like we can get like funding soon to that. And I will stop here, and thank you so much for your attention.